Hey everyone, and welcome to This Explainer. I am an independent biotech postgraduate researcher, and today we're unraveling a massive scientific detective story right here from the lab. What if I told you that one of the most incredible discoveries in blood disorder research, like something that could literally rewrite the future for millions of people, came from a technique that doesn't rely on making a single prior guess?
Okay, let's dive into this because the sheer scale of the human genome is, well, it's absolutely mind-boggling. Inside almost every cell in your body, you've got about 3 billion DNA letters making up your personal instruction manual. When we're looking for the cause of a complex condition or a potential cure, we aren't just looking for a needle in a haystack. We're looking for a specific microscopic needle in a mountain of them. How on earth do we locate one single genetic dimmer switch that controls a disease among 3 billion individual letters, especially when we don't even know exactly where to start looking? We need a tool that can just look at everything all at once.
Which brings us to Section 1, the genomic search engine, scanning the blueprint of life. Enter GWAS, which stands for Genome-Wide Association Study. I want you to think of GWAS as a massive, ultra-powerful search engine for our DNA. In my independent lab research, I rely heavily on this exact concept. Instead of just kind of guessing that a certain gene might be responsible and only looking there, I use GWAS to scan the entire genome. We're looking for SNPs, single-letter changes in the genetic code. By comparing the DNA of thousands and thousands of different people, I can clearly see which specific genetic letters are associated with particular traits, like naturally high fetal hemoglobin levels. We essentially just let the data speak entirely for itself. So let's break down exactly how this methodology acts as my blueprint in the lab. Step 1. We start with highly advanced sequencing equipment that identifies known genetic markers within patient DNA samples. Step 2, we scale this up massively, scanning millions of these single-letter changes, or SNPs, across the entire genomes of our study participants. And finally, Step 3, and honestly, this is the hardest part, we take that absolute mountain of raw genomic data and we rigorously filter it. We absolutely have to separate the random genetic noise from the true, undeniable disease associations. Moving right along to Section 2, visualizing the statistical skyline, finding the truth in the data. You know, in genomics, you can't just be pretty sure. You have to be absolutely mathematically certain. Because we're running millions of tests at once across the genome, the chance of finding a false positive is just huge. So to prove that our genetic findings are actually real, my lab strictly adheres to this extreme statistical threshold, a p-value of less than 5 times 10 to the power of minus 8. So put that into perspective, that is an incredibly tiny number. It means the association we're seeing is so ridiculously strong, it's almost statistically impossible to be a mere coincidence. And this brilliantly illustrates how we actually look at that math, the Manhattan plot. We call it that because when you visualize all this genomic data on a scatter plot, it literally creates a city skyline. The bottom axis represents all our chromosomes, and the vertical axis represents that super strict statistical significance. When you see towering statistical peaks shooting up into the sky on this graph, they're pointing directly to significant genetic truths. Those skyscrapers are basically the genome shouting, "Hey, look right here." Let's look at our timeline and see how this builds our understanding of blood disorders. Pre-2008, scientists were desperately seeking the master regulator of fetal hemoglobin, basically the key to potentially curing conditions like sickle cell. Then, in 2008, researchers ran a GWAS and saw a legendary skyscraper breakthrough on chromosome 2p16. The statistical significance of this peak was less than 10 to the power of minus 15. I mean, it absolutely shattered our strict threshold. Today, our entire field is focused on targeting this very specific genetic switch to permanently cure blood disorders. This brings us to Section 3, the non-coding dimmer switch, zooming into the BCL11A gene. The sheer elegance of human biology is just amazing here. The discovery from that skyscraper peak wasn't a mutation inside the coding part of the gene itself. Instead, it was an erythroid-specific enhancer. It was hidden deeply within a non-coding region called the second intron of the BCL11A gene. You kind of have to think of this non-coding DNA not as junk, but as a brilliant precision dimmer switch. And the most mind-blowing part, it controls the expression of the BCL11A gene exclusively in your red blood cells. So, the crucial point is understanding this relationship. If the BCL11A gene is the heavy machinery inside the factory, the thing actually doing the work of turning off fetal hemoglobin, the enhancer in that second intron is the hand on the control panel. It's the precision dimmer switch controlling that machinery. When this specific switch is flipped or altered, something incredible happens to our hemoglobin production. It literally opens the door to turning healthy fetal hemoglobin back on. Which leads us nicely to Section 4, Cures for Diverse Populations, translating data into localized treatments. Think about this comparison for a second. Historically, most of these massive genomic studies were conducted predominantly in European populations, which unfortunately means we have really limited global representation in the data. The switch might act slightly differently depending on your exact genetic background. That's exactly why the core mission of my lab is laser-focused on Indian communities, stretching from Maharashtra all the way to Odisha. These tribal regions and communities possess highly unique genetic diversity. It's a massive beacon of hope for the global community, but we absolutely have to do the localized work to understand it. My goal is to ensure absolutely no one is left behind. We're out here studying these unique population variants, discovering exactly how this molecular dimmer switch might differ globally. Because while the world has finally found the where on chromosome 2, the how for every specific population remains a puzzle that I am determined to solve. We use advanced, independent lab techniques to translate these localized, raw data findings into potential real-world patient treatment plans. We literally must ensure that the genomic cures of tomorrow are perfectly calibrated to work for Indian cohorts today.
And finally, Section 5, Decoding the Future, applying the map to your DNA.
So we've identified the microscopic needle, we've mapped out the BCL11A enhancer, we understand the unique genetic variations across diverse populations. But what happens when we take the next huge leap? What happens when we take powerful biotechnology like CRISPR and actually reach into the living cells of a patient to physically flip that dimmer switch ourselves? How do we turn a statistical peak on a computer screen into a literal biological cure for a devastating blood disorder? Well, I'm not giving away the final answer just yet. The transition from raw data to clinical reality is definitely the most exciting part of this entire genomic mystery. If you want to dive deeper into the science with me and see exactly how we're turning these statistical peaks into real-world CRISPR cures, you can explore more and follow my research channel via the link in my bio. Thank you so much for exploring the blueprint of life with me today.